Good afternoon and welcome to the joint meeting of the Virtual Nitrogen Centres under the Indo-UK Newton-Bhabha Fund. I am Raghuram, co-leading the News Nitrogen Centre with Sutton and other institutions in India and UK. We are in a historic place. Most of you who have been already working with the IRI would be aware that this is a part of a complex called POSA complex, which hosts the Indian Agricultural Research Institute. The building we are sitting in is the National Academy of Agricultural Sciences, and next door we have the National Bureau of Plant Genetic Resources. So this whole campus is a very historic center, and also the place from where the Green Revolution of Indian agriculture had started years ago. And this complex and the NBPGR are also symbols of India's international cooperation. Of course, one could say even the Green Revolution of India is part of the international cooperation with the US at that time. So in that sense, we are in a place that has been long a symbol of international cooperation. And this meeting and our nitrogen center that I am leading and the Indian nitrogen community has been a part of a long process of engagement of over a decade under the Society for Conservation of Nature, started by Professor Raghuram, who was the head of the plant science department here, plant physiology department. The Indian Nitrogen Group was established in 2006, under which there was an attempt to bring together interdisciplinary talent in all aspects of the Indian nitrogen cycle, the biogeochemical cycle, to understand the reactive nitrogen cycle and quantify it. More on that later. That gives us a sense of where reactive nitrogen stands in India, where the problems are, why agriculture nitrogen use efficiency is important. And many of our partners, in fact, from more than one center actually, have been part of this Indian Nitrogen Group, and it gives us a great sense of pleasure that the long process of collective understanding and team cooperation has also helped us in this international set of centers. And this is also the first center, first opportunity where all the 4 virtual nitrogen centers are able to meet together in one place. The CINTRIN, the INEW, the IUNFC, and the news.
So thank you, Raghuram, and I don't need to say a great deal other than to think about what are our goals for this meeting, and briefly thank, of course, DBT, BBSRC, RCUK, and the Newton-Bhabha Fund for actually making the whole thing possible. So we're really happy that you're here with us, and we hope that in this session, also today, we can actually show to you why the world should be caring about agricultural nitrogen. For me, I put it into money. I started as an environmental physicist and a botanist looking at fluxes, and then I at some point started looking at impacts, and then at another point I started trying to generalize why anybody should care, and I found out that money is the reason actually people care in the end. You can value ecosystem services and get really big numbers, but let's just put it like this, that India is spending around $7 billion a year on its fertilizer subsidies. The total input is around $10 billion, and actually the losses are even bigger than that because you're losing from biological nitrogen fixation losses as well, and from manures. So you're losing over $10 billion a year of fertilizer value nitrogen in India, and this program aims through a whole range of means to reduce that number. So I think that's a really laudable aim, and of course the environmental benefits for human health through air pollution reduction, greenhouse gas mitigation, water quality improvement are going to be even so much bigger than that, whatever the value that we estimate. So that, I think, is the agenda, and of course then there's all the tools for doing it, with the focus on biological nitrogen fixation, plant nitrogen use efficiency and genetics, agronomic perspectives. So that's our challenge. In terms of, we've got these 4 centres you've seen on the board. They're working on different things, and we want you, as you come together, to hear what you're doing, and of course introduce everybody to get to know each other and build something of a community.
So the Newton Fund is UK's flagship bilateral program, which was initiated in April 2014, and it aims at two objectives, primary and a secondary one, which is to promote economic development and social welfare, and also to develop research and innovation capacity for long-term sustainable growth. The total UK funding is £735 million up to 2021. I'm sorry, it's not a 20-year yet. We were supposed to be till 2019, and there was later there was a review which expanded the program in terms of the money. So from a 375 million, we were increased to 735, and from 2019 we got an extension until 2021. It is largely managed by the UK Department of Business, Energy and Industrial Strategy, that is the base. And a significant feature of this program is that it is match funded. So we're talking about partnership of equals, where we have both the UK and the partner country putting in the same amount of resources or money into the partnership, into the programs. The key principles of Newton Fund are, of course, partnership, capacity building. We aim to tackle global challenges, and we support excellence in research and innovation. Newton Fund is based on a very fair, competitive and open, transparent system, and we aim to support the best projects and the best scientists out there. Here's a list of the partner countries. Currently we are partnering with 18 countries across the world, and Jordan and Peru are the newest members of Newton. You would be happy to note that India, China and Brazil hold the largest portfolio for Newton programs. So every Newton country has a significant name to its partnership, and in India we call it the Newton-Bhabha, named after the famous scientist Dr. Newton-Bhabha. It was initiated in November 2014, and again, as I mentioned, it's a partnership of equals. So from the UK side, there's a total investment of £104 million, and equally supported with the same amount by India through resources and skills and money.
The RCUK India team was established in 2008. We are based at the British High Commission, and we're 6 members. Effectively what we do is we represent the UK research councils here in India. Our job is to help them work with partners and Indian funding agencies to make sure that we can come up with bilateral research programs that are academically excellent, that will have high impact, and basically help to develop a partnership that is more than what we could hope to achieve by just working from the UK. So we've built a very strong relationship with many partners in India. A few of these will obviously be very familiar. The Department of Biotechnology, who I think is our biggest partner now, I'm pretty sure of it. ICAR's up there, who we work with in Skipford. We also have a number of partners representing social sciences, physical sciences, and DAE as well, Department of Atomic Energy. And in terms of the UK side, as I said, we represent the 7 research councils, which are listed there. We've also worked closely with colleagues in Telford, colleagues in the Newton Fund, Innovate UK, the EU to develop social science programmes, and from April 2018, the research councils, RCUK, will actually be changing into UKRI. UKRI is basically the 7 research councils plus Innovate UK and an organisation called Research England. When that happens, we will basically be working even harder on developing collaborative relationships with India that represent the research councils plus those organisations plus others. So it's a very exciting time for us. Our portfolio covers a range of different topics, as you can see, ranging from smart energy, renewables, through social change, civil nuclear, advanced manufacturing, digital economy, food security, agri-tech, water and climate. These topics aren't random. One of the things that the office does is work very closely with our partners here in India, our funding partners, to make sure that the programmes that we're funding are aligned to Government of India priorities. The reason why we do that is for what Professor Sutton was actually just mentioned, is to help with longevity and to make sure again that the programmes that we're funding will be supported and will lead to some very good impacts.
We talk about CINTRIN being a truly translational centre, and that's because we take our thinking all the way from basic developmental biology and physiology in model species through crop breeding and transgenic strategies to field scale performance, technology and extension. Here we can see the 6 work packages of the CINTRIN project. The first one is about the basic research on biological nitrogen use in models. The UK leader on that is Ottilie Liza from the Sainsbury Laboratory, and the Indian leader is Manoj Prasad from NIPGR. The work package 2 is defining optimal cereal use on key cereals, and that's led by Howard Griffiths at the Department of Plant Sciences and the University of Cambridge, and Rakesh Shrivastava at ICRISAT. And work package 3, where we're translating the knowledge we have of nitrogen via genomics-led pre-breeding transgenics, is led by Alison Bentley and Emma Wallington of NIAB and Rajiv Gupta of ICRISAT. Work package 4, we're looking at field scale evaluation and optimising nitrogen allocation, and that's led again by Howard Griffiths and Roger Sylvester Bradley of ADAS in the UK and Parveen Juneja of Punjab Agricultural University. The work package 5, where resources for breeders and farmers, led by myself and Sashi Shende of Kisan Hub, Santosh Deshpande of ICRISAT and Vrinda Pal Singh of PAU. And the final work package is to expand the network, led by Mariana Fazenda at Cambridge University and Rajiv Gupta at ICRISAT. So in work package 1, we said we would exploit the genomic resources in models and look for candidate genes in wheat, sorghum and pearl millet. In work package 2, we're looking at where that nitrogen goes. We're looking at variation for physiological nitrogen uptake and allocating how the nitrogen is allocated to the grain through the use of nitrogen and carbon isotopes. And the idea is twofold: to identify parallels with the developmental idiotypes described in work package 1, and also to look for natural variation in nitrogen partitioning for growth in a range of cereal species using resources available in the UK and in India. Work package 3 is about translating that knowledge some of which we already have, and the new knowledge that we're generating into the project through to breeding material. And this slide just shows the reason why you need to do it through genomics-led breeding, and you really need to do it through some form of genetic engineering, simply because of the time it takes to get there. This 20 years has been mentioned a couple of times already. We don't have 20 years. It'd be great if we'd be able to do something significant in 2. We were going to develop next generation germplasm for breeding in wheat, pearl millet and sorghum. We were going to develop a range of phenotyping tools and techniques, and we were going to validate gene candidates via transgenics and gene editing. Work package 4, so this is where we're moving to the field. And so one of the things that we introduced into this project, is led by Roger Sylvester Bradley of ADAS, is to try to identify varieties which had a high yield under a lower optimum level of nitrogen fertilizer. So we'll be trialling the pre-breeding germplasm that we've identified in the earlier work packages with varying nitrogen inputs in field experiments in the UK and in India to validate the nitrogen use type, and also to select for high yield, low optimum nitrogen types in the field. So these high-low lines will be identifying the threshold above which adding more nitrogen does nothing for your yield or your economic yield. One of the other exciting things we've been doing, and I was very pleased to see the upcoming call on data, because one of the things that we've been doing in CINTRIN is to see if we can use a cloud-based software platform to hold our data. This is basically what it looks like, and Vasil, I'm not going to attempt your full name, Vasil is going to talk to us later a bit more about the platform. In link to this, in work package 5 and work package 6, led by Vrinda Pal Singh in Punjab Agricultural University. There's been the development of a leaf colour chart using optical sensor data and input and soil balance modelling. And this enables farmers by a simple visual method to identify how much they need to put on. So rather than putting on the normal level, the 150 kilograms per hectare, whatever it is that they would put on, it says according to the colour of your leaf matched by this chart, you can probably get away with less. And
I'm going to introduce the INEW to you. This is the Indo-UK Center for Improvement of Air Use Efficiency in wheat. So in India, wheat is grown and wheat area is divided into 6 zones. So that is the Northern Hills zone, and the major area which is contributing to production is Northwestern Plain zones and Northeastern Plain zones. And some of the area is in Central India, that is Central zone. And some of the other quality wheat, that is Dicocum and Durams, are grown in South India, and Durams are mainly grown in Madhya Pradesh, that is Central India. So coming to the production, we are producing almost 1/7 of the global production, that is we are nearing up to 100 million tons. This year we have produced 97.4 million tons of wheat, and to produce so much of wheat we are consuming a lot of nitrogen also. So this comes the importance of nitrogen, and we are losing in terms of monetary terms around 2000 crores of rupees on this, on wheat only. Coming to the partnership in the INEW, so we have chosen the partners based on their strengths, and they are divided into assets like this. And the program is led by IWBR Karnam, which is known for testing and release and delivery of wheat varieties in India for last 50 years, and IRI, which is the main center working on wheat research and delivering major varieties for the region. And then NRCPB, they are known for biotechnological research in agriculture, so they are involved in all high throughput sequencing programs and all, so they are experts in that. And then NBBGR, which is involved in handling data management part of the program. And then BISA is involved in precision delivery testing. So here they are conducting an experiment in delivering specific doses of nitrogen to the root zone at specific time intervals. So we want to identify the package of practice for the farmers. So coming to the objectives, so we want to exploit the mutual expertise of the collaborators, for example complementarity, and then align the resources like UK has the genotyping platform, and also we are also capable of genotyping the centers, and then we are well versed with many of the centers which are located all across India involved in wheat research. And then we want to use the shared platforms, like, so we will use the common protocol for any use efficiency screening, and then we are collectively pulling the germplasm also. So we have got 200 germplasm lines from UK. They are derived from the vaccine collection lines, and in India also we had the legacy of as it was mentioned, nitrogen group was started much before, and some of the experiments was conducted in that, and the genotypes derived from those experiments were used in this project for the further study. And then identifying the tools, lines and tools for high-end use efficiency genotypes and markers here. So we are going for JIVAs and association mapping of the lines for probable identification of genes and QTLs involved in use efficiency. So I want to highlight that the major thrust area of this project is looking at the genetics and identifying the donors for NU efficiency important traits. And also the major one of the highlight is training the capacity building of the students and also the project scientists involved in the project. So there is a lot of budget involved in this, and both the countries are working together in conducting these trainings and some of the exchanges in terms of young scientists going there to the UK labs and getting themselves trained in the high throughput areas is going on.
The sort of bulk of the project is actually on phenology, comparing phenology with nitrogen use efficiency. This concept comes from Simon Griffiths. This is a photo taken, I think, at Karnam earlier in this year when we visited the field plots in March. And basically, it's how can you adjust the timing of the development or select the time of development to make the best use of nitrogen. What we're doing is selecting sister lines based on adaptation to the UK and India. Drawing relationship between the phenology development and NUE and vacuum QTLs using these established markers. For the source of variation, we use the Watkins collection of land bases, and actually my colleague Teal Cohen will talk about this a bit more tomorrow, but it's a collection of land bases from the 20s and 30s, from various countries including India. I'm afraid it's very largely sort of the old British Empire, because that's what they did, they sent people out to collect things, to send back wheat. But very, very useful source of variation. As an additional source of variation, we've also used some of the magic lines from NIAB. I presume someone from NIAB will talk about magic at some stage of the conference. If we could talk about it later. But this is effectively what we used to call a composite cross. You take 8 varieties and cross them in all combinations to get a very high degree of recombination variability. So far we've genotyped all the material from the UK at Bristol. There's also genotyping being carried on with the same system here at MBPGR, and they're also doing some RNA-seq studies. I presume that PK Mandel will talk about this later on when he's talking in the meeting. Work carried out here in New Delhi. The Indian project started 6 months after the UK project. So it means that in fact this was very useful because we can alternate our field trials. We're working with spring wheat. In the UK we plant in March and we harvest in August. In India they'll plant in October and they'll harvest around about April. So it means that we can carry out a UK experiment, take some information from this into the Indian project, and then have a visitive process. And in fact the first 2 years field trials we use the same genotypes in the UK, we'll probably use the same ones in India for the second year. But it's been very useful to have this slight sort of delay in the Indian partners so that we have this overlap. We've got a very big training programme. We decided that training capacity building should be a major part of it. So we put in for we have a grant for 2 components, talk courses and exchanges, budget of about £150,000, and that's about 10% of the UK budget. That's a substantial amount of money. Courses, this is the course that Tina mentioned earlier. We actually ran it as a joint course with the 2 programmes. They got something like, this was run at Raghuram about a year ago now. They got over 70 applicants and selected about 26, mainly Indian academics and students, one from the company, 2 UK, and say half of those people were from CINTRIN, half were from INEW. And that was a very successful course. We've run 2 courses in the UK. We ran them this summer. We ran one on wheat genetics and market development, and then we ran a 3rd course on phenotyping and phenomics, about 12 participants, Indian students, Indian researchers, UK students.
Good afternoon. The Indo-UK Nitrogen Fixation Centre is a collaboration between 7 laboratories in India and 3 in the UK. In the UK it's led by Philip Poole from the University of Oxford, and his co-PI is Ramachandran. In India, it's led from Indian Institute of Soil Science, which I represent along with my colleague Dr. Mohanty. In this project, we have 2 principal crops. One is pigeon pea, symbiotic nitrogen fixation, and then endophytic nitrogen fixation in rice. The 3 objectives, or rather the work packages which we have, are improving pigeon pea rhizobium symbiosis for high DNA potential under stress, develop engineered rhizobia with phosphate solubilizing ability, and then look at rhizobial endophytes on how to use them for improving nitrogen nutrition of cereals,
and reduce fertilizer nitrogen dependence. India is the center of origin of pigeon pea. It is the most important rain-fed crop in agriculture. As you know, 50% or more than 50% of Indian agriculture is rain-fed, and this is the principal legume. It has got deep roots, nodulates into a deeper area, and pigeon pea represents about 15% of the pulses crop in India and about 17% of production, and almost about the average yields are only about 900 kilograms, 848 kilograms to be precise, but there's a large deal of variation. You can look at the huge variation between that. And nitrogen fixation has been estimated only perhaps once using nitrogen abundance method, not using isoabundance but using labeling. And we are now going to use what is called as nitrogen abundance method to isoabundance method. And because it is grown in summer and rainy season, when temperature is very high, there is drought, and usually used by marginal farmers. So it's a low input farming. And usually most of the Indian soils which are in the tropics have got low soil organic carbon, that means low soil biodiversity, and no ability to support nitrogen fixation. So what are we going to do? What we thought was that we'll look at a variety of genotypes of pigeon pea which are currently being grown, and we took all the three major soils. Pigeon pea is grown in north, is grown in the center. North is the insectosols in the north, and then the vertisols in the center and alphazols in the south. So we're going to look at all the three soils and look at various genotypes and their ability to nodulate, isolate a number of strains, and then look at also other plant growth promoting rhizobacteria. There are a lot of other rhizobacteria in the soil which enhance nodulation. These are plant health promoting bacteria which will help nodulation. And then at Oxford we are going to actually, we have already done the 6-SRR sequencing of number of rhizobia, and will present those results. But we're going to look at the entire genome and then do a kind of a pan-genome analysis of the entire soil and rhizobia, which Phil is going to do that. And most importantly, the field measurement of nitrogen fixation using isotope-abundance method. We're going to do it at all the three soils. And then we also work on soil metagenomics, looking at the entire genome of the soil and look at what are those situations where pigeon pea symbiosis is improved. So that's about the first module. The second one is on enhancing plant growth and BNF by engineering rhizobia to solubilize phosphate. Phosphate supply is very critical, as you know. So Naresh Kumar is going to use various methods of whereby rhizobia which hyper secrete 2-ketoglutamate acid or citric acid and oxalic acid and others. So that in diverse soils you can have, and I have seen some of the results in soybean, they are really fantastic. That rhizobia which can also solubilize phosphate bring about dramatic improvement in nodulation and fixation. In mung bean, now we want to do that in some other crops. So this is the expression of enzymes for secretion of various acids, and then grow them in soils under limitation and access. The idea is to get some strains which can, across various soil types, solubilize phosphate. The third objective is characterization of rhizobial endophytes of rice and their mechanism of infection. So Dr. Maitreya Dasgupta has got a number of strains of rhizobia. Now, because historically legumes have always been used as a green manure before rice. So always the Indian rice fields have always seen a lot of legumes. So there are a lot of rhizobia already there, and although the population declines when rice is grown, so some of those rhizobial strains have got an ability to infect rice but not form nodules. So they are intercellular colonization numbers, and she's got various kinds of strains of rhizobium which infect rice but don't fix nitrogen. And there are some which fix nitrogen with Arachis hypogaea, which is a candidate legume which is being used here. And some of those Arachis hypogaea strains which are able to fix nitrogen, those rice strains are able to fix nitrogen in the case of rice. So what are the impacts? What we now finally hope to do is to deploy effective combinations of pigeon pea genotypes and rhizobial strains to maximize BNF, and develop rhizobial hyperphosphate solubilizing ability to improve BNF, and understand how rhizobia colonize cereals and engineer ammonia release to plants. There's a big component of training of young scientists. One of them has already been trained, others are on way. And then promote, and there is also an Oxford India Center for Sustainable Development, which is also part of this program. And we have just had a meeting in Oxford. And then we're going to look at how to promote legume usage in meeting the Sustainable Development Goals, and then an outreach to farmers and public in the form of using these improved rhizobia for inoculum production.
Thanks for everybody for listening. I'm just going to sort of summarise really where we've got in that overall work packages in the last 12 months or so. And as D.L.N said, we just had our recent meeting, so it's a sort of convenient time to sort of reflect on where we've got to. Just to repeat though, if we look at pigeon pea, just to remind ourselves that it really is such an important crop across India in dry environments. It's really important. So D.L.N's already gone through the basic work packages, and the first one was really about selecting optimal pigeon pea rhizobium combinations, because clearly if you want to get the maximum yield out of any legume, it can be very important to inoculate with the right rhizobia. In many cases, of course, people don't inoculate, they don't maximise the yield, but where it's available, inoculation can dramatically improve the performance of the plant and the yield. So clearly, what would we want to do? Remembering that pigeon pea is important in rain-fed agriculture, we really want to try and select the best rhizobia for biological nitrogen fixation. So to get the maximum yield, to get the right strain together with the right rhizobia. And so what does that really involve? It involves a lot of high-throughput screening, actually screening different pigeon pea varieties and the rhizobia you get out of the nodules, and testing how much, what yield of plant you get, as well as then going on to measure things like 15N, actually looking at the amount of 15N2 nitrogen fixed. And then, of course, trying to add some science on this to understand the basis for that, to actually start doing a lot of genome sequencing of those strains, and also then to do metagenomics of what's in the soil, what microbes are there at the same time, and of course going back to field testing of varieties and strains we work on. I should say, in this particular package, so what's happened now, the Indians have been very busy. They've done screening of large numbers of lines, isolating rhizobia out of the nodules, looking for the strains which are giving the best rates of nitrogen fixation. So that's been a big project in 3 different sites, and then multiple soil types over those sites. So that work is very much in progress. The second work package is this one of actually looking at nitrogen fixation and how you can improve it by solubilizing phosphate and increasing plant growth. And that's really been going on at the moment at the level of actually looking at the enzymes that will do this, actually trying to select systems and understand biochemistry. And in the future we're going to be able to take those and adapt them to new strains of bacteria. So, and then of course assess that for actual growth in the field if we actually use those strains. Work package 3 is this characterisation of diazotropic rice endophytes and the mechanisms of infection. One of the big areas that isn't just within this work program, there's a global series of objectives now, is to try and get endophytes which may fix nitrogen in association with a variety of different cereals, and to be able to maximise the yields of that, and also to impose control mechanisms between the plant and the microbe. I won't go into a great deal of analysis of that, but there is a program here, particularly in Maitri Gupta's lab, to actually look at bacteria which naturally colonise rice, and which also appear able to fix nitrogen. And so we've been looking at the genomics of those strains and trying to understand what basis there would be for getting nitrogen fixation from those bacteria directly in association with cereals. And that involves a number of things we want to do, both develop new strains of bacteria which will fix nitrogen in association with cereals, but also to get them to actually secrete ammonia. So what basic progress overall, as I said, we've had large-scale screening of rhizobia have gone on in India on pigeon pea. We've done a lot of genomics of marker strains. A lot of work has gone on with phosphate solubilization in India. The metagenomics pipelines that we need have been refined, and we started working in detail on genetics of ammonia secretion to identify key mutations. So there are actually only 3 characterized strains at the moment of pigeon pea rhizobia, and none of them come from India. 3 strains, not 300, not 3,000. And finally, it's actually looking at how you can engineer ammonia excretion by bacteria to actually, so that we can get, well, if I start from scratch, the first point is, if you want to get nitrogen fixation by any bacteria, you need to get a carbon supply out of the roots of plants. That carbon supply from the plant can then drive nitrogen fixation, which you have to be able to engineer at some level nitrogen secretion by the plant, or select for natural variation within bacteria. And that in turn would then obviously going to need nitrogen assimilation.
So I want to just say why India first off, and here's an interesting map. This is the map from space of ammonia concentrations of the world. It isn't a perfect map, but it's a rather shocking map. So this is the depth of ammonia in the atmosphere. And you can see North India shining out 100 miles compared to the rest of the world. There's a specific question about why is ammonia concentration not so high in China when the emissions, we think, ought to be high. But anyway, this is a clear driver for interest, not just of where this came from through agricultural losses and improving agriculture to reduce emissions, but also in terms of what the effects will be. So that's just a starter. And I'm going to mention three objectives. The first one, quantifying relationships between European people measuring losses. I'll say a bit about the losses, looking at all available nitrogen sources and going towards a nitrogen budget for India. And consider the effect of possible policies. I think this is interesting for the discussions tomorrow. And so 2015 already had the neem-coated urea policy. I will declare this to be the biggest and most significant policy decision on the nitrogen cycle in the world in the last 10 years, because there's at least 1.2 billion people in India, and all of the urea is now coated with neem oil, which reduces denitrification, we think. What about a future policy? Neem plus policy, what would be neem plus? And what about better manure recycling? How would one frame it in a policy? So those are sort of questions we have. To go then into component 2, quantifying those different fluxes, emphasis both on the plot scale and comparing genetics, but also on measuring the emissions and going towards full nitrogen budgets. So neem-coated urea, really good thing, reducing denitrification losses as N2O. Less greenhouse effect from agriculture. However, we've got the neem-coated increasing ammonia emissions. Isn't surprising, because what the neem is doing is reducing the rate of nitrification from ammonia to nitrate, meaning that it stays in ammonium form for longer, hence increased ammonia emissions. So I would say neem plus would be, imagine a nitrification inhibitor like not neem together with a urease inhibitor, be it NBBT or whatever. And for India, for example, the approach may be produce it in the urea melt commercially by Indian manufacturers. It could be done at low cost. To say a bit more briefly about components 3 and 4, we're onto the village scale here, working at farms, and Sander Bandapada will present about this tomorrow, with a focus on tracing the manure following excretion from small farms who have 1 or 2 cows, and saying, can covered manure storage, how much is that saving nitrogen in the system? How much can immediate incorporation reduce losses? So really very practical at the field level to get all those nitrogen sources maximized. Component 4, going into the upscaling for India and comparison with China. So the challenge of improving nitrogen use efficiency. This is a graph, old graph from Tapan Adja, who I think is here, showing partial factor productivity for nitrogen decreasing over the last decades, and as nitrogen inputs have gone in, the nitrogen efficiency has gone substantially down. So this is a real challenge for us to turn that around, and some calculations even we've already started with is if India was to go for a 20% increase in national nitrogen use efficiency, as a fertilizer value alone, it'd be saving 1.5 to 2 billion pounds a year worth of fertilizer resource. I think the challenge we face is to now then pin down those different options of how much can each option achieve and how much can that be contributing to that. So let's just to finish, we are looking at key advances in nitrogen use efficiency, looking at capability increase to measure full nitrogen fluxes, improve our methods, demonstrating the extent of synergy between agronomic approaches, current nitrogen use efficiency, looking for better valuation of all organic nitrogen sources, not forgetting every source, and looking towards then this national picture of nitrogen management towards a join that picture for climate, environment, food security together. And the bottom line message is then more profitable farming with less pollution, and going all the way then from the genetics that Raghuram has mentioned up to the policy application.